Hi, welcome to Microbiology Mentor. In this session, we will learn about the type of vaccines. So let's see what is a vaccine first. A vaccine is a biological product that can be used to safely induce an immune response that confers protection against infection or disease on subsequent exposure to a pathogen. Now let's see the types of vaccine. Classically, the vaccine types were either live or killed on the basis of the nature of how it was produced. But with the introduction of various other platforms, other types of vaccines are also given name like subunit vaccines, toxoid vaccines, polysaccharide and conjugated vaccines, recombinant vaccines, messenger RNA vaccines, viral vectored vaccines and other miscellaneous types. So let's see each one by one. So first, live attenuated vaccines. As the name suggests, it contains live pathogens which have been attenuated or weakened. So they have got the potency to replicate and to trigger an immune response, but it does not cause a disease. The classical example of live attenuated vaccines includes measles, mumps, rubella, yellow fever, influenza, oral polio, typhoid, Japanese encephalitis virus vaccine, rotavirus vaccines, BCG vaccines, and varicella zoster vaccines. Usually, the live attenuated vaccines require a single dose, but there is an exception to this rule where the oral polio vaccines requires multiple doses to mount an immune response. Let's see what is a killed or inactivated vaccines. Here, the whole microorganism has been killed and it has not got the capacity to replicate because it is no more live. So multiple subsequent doses are required to generate or to build an immune response. The example include whole cell pertussis vaccine, polio vaccines, the injectable one, influenza vaccine, Japanese encephalitis vaccine, hepatitis A vaccines, and inactivated rabies vaccine. Now let's see subunit vaccines. The subunit vaccines contain the pieces of pathogens, and it can be of many types of subunit vaccines like purified proteins, recombinant proteins, polysaccharide and peptides depending on the nature of the peptides. So the example of subunit vaccines includes the pertussis vaccine, influenza vaccine, hepatitis B vaccine, meningococcal vaccines, pneumococcal vaccines, typhoid and hepatitis A vaccine. Now coming to the toxoid vaccines. So the toxoid is produced by harvesting the bacterial toxin, changing its chemical nature, which is usually treated with formaldehyde to convert a toxin into a toxoid. So toxoid are able to induce an immune response and neutralize the toxin, but it fails to produce any clinical manifestation. The classical example of toxoid vaccines include the diphtheria toxoid and tetanus toxoid vaccines. Now coming to the polysaccharide and conjugated vaccines. The polysaccharide itself has a poor immunogenicity because it induces only the primary immune response and fails to induce the immune memory. So it is usually conjugated with a carrier protein. When it is chemically attached with a carrier protein, it is able to activate T cell response and induce antibodies against the desired antigen, and also at the same time it is able to induce an immune response. The example of protein polysaccharide conjugated vaccine include the Haemophilus influenzae type B vaccine, the conjugated pneumococcal vaccine, conjugated meningococcal vaccine, and conjugated typhoid vaccine. Now coming to the recombinant vaccine. Recombinant vaccine is usually made using a gene from the disease causing pathogen. These genes is recombined into the genome of a cell, which is usually taken as yeast cells. The yeast cell multiply and the copies of the desired gene is also multiplied and produces the protein which produces and can generate protective immune response. The example of recombinant vaccine include the hepatitis B vaccine. Now coming to the nucleic acid vaccines. The messenger RNA is used to deliver the genetic information in the form of nucleic acid to make the specific viral protein. This mRNA is of the nature because it is getting easily destroyed. So before introducing inside the cell, it is protected by a lipid nanoparticle. Now once it is lipid coated, it is injected inside the susceptible host, it generates viral protein, which then can be presented to T and B cells in the lymph node to generate an immune response. The classical example of nucleic acid vaccine include the SARS-CoV-2 vaccines. Now, what is viral vectored vaccine? A viral vectored vaccine is also using messenger RNA to code for the protein, but the method of the transportation into the susceptible host is rather different. Here, what they use a adenovirus, which is a harmless virus, to introduce the protein into the host immune cell. The immune cell then create protein from the messenger RNA and trigger an immune response like normal. The example of viral vectored vaccine includes the Ebola vaccine as well as the AstraZeneca COVID vaccine. Now, apart from these, there are other miscellaneous type of the vaccines which include the bacterial vectored vaccines, where the gene of a pathogen of desired is introduced in the bacteria and it is acted as a vector and introduced in the human cell to trigger an immune response. This is under experiment. Other type include the virus-like particle, where the virus-like particle mimicking the virus can be introduced to trigger an immune response, like in case have been tried in human papilloma virus. Apart from that, outer membrane vesicle, which is made from gram-negative bacteria outer membrane and coated with the bacterial pathogens, can be used to trigger an immune response in a host, and this is used for group B meningococcal vaccines. With this, we come to an end of the topic of today. Thank you.